Hey, what is going on? It is Thursday, March 10th, 2022, and as you might have noticed yesterday, I did not put up a video. I was pretty busy with what I was doing, and to be frank, I kind of let a lot of the day slip ahead of me, slip by me, which I plan not to do anymore. But yeah, yesterday was a hodgepodge of things that I was trying to take care of. But today I'm back at it, and I am now talking about phosphorylation. Okay, so I had made quite a jump because the last time I was talking about the transition phase of between glycolysis and the Krebs cycle, and how the products of the Krebs cycle the products of the transition phase will eventually go into the Krebs cycle, and also they regulate the transition phase as well. So according to Le Chatelier's principle, products will drive their production of reactants, and reactants will drive the production of products if system is in chemical equilibrium. Okay, so just go look that up and whatnot, and that's essentially in a large way how the transition phase functions. And just to remind you what the reactants and the products in the transition phase are, the reactants, remember, we get pyruvate, we want coenzyme A, and we want oxygen, right? And what we get out of that as waste, we get carbon dioxide, because pyruvate gets converted to a 2-carbon molecule. We get acetyl-CoA, which is the final product, right, of the final primary product of the transition phase of glycolysis, which eventually goes into the Krebs cycle. And we get NADH, because we get NAD as one of the reactants as well, which I neglected to mention. And all of those go into the Krebs cycle and electron transport chain downstream, which the end result is 34-ish, even more, 34-ish ATP, right? That's the whole goal. You got to remember the goals in biochemical reactions, right? Because a lot of times it's easy to forget what the whole point of all the reactions are. So today I'm going to take a huge step back, because I've been trying to get to step 3 of glycolysis actually, and I realize again I'm jumping around and out of order, but I do want to get to phosphofructose kinase and talk about the allosteric regulation of that, and how phosphofructose kinase essentially, remember, now I wanted to take a brief detour into what kinases are. I mean, by the time you get to this point, you probably know what protein kinases are anyway, but I just wanted to recap that. So basically, we need to, in order to recap that, I've been also liberally using the term phosphorylation, because I'm kind of taking it for granted that everyone kind of knows what phosphorylation is when they watch these videos, but just to take it up to the very basics, right? Phosphorylation is kind of the key mechanism by which all of biochemical activity occurs, right? And remember, biochemical activity, as in all activities in chemistry, are primarily driven by energy considerations, right? The lowering of your energy state, because everything essentially wants to go from high energy to low energy. So I'm not going to talk about the Gibbs free energy here, but most chemical reactions are considered, the direction of the reactions are considered from products to reactants to products based on their Gibbs free energy when a system is in equilibrium. So let's go back again, focus on energy. So phosphorylation is the core energetic indicator reaction in biology and biochemistry, right? So it's basically the conversion of we basically want to add, we want to take an ATP molecule, right? That's a highly energetic molecule. Remember, ATP is adenosine triphosphate, and we want to take this ATP and add a phosphate group to something else, or we want to take an ADP and add a phosphate group to ATP. Add a phosphate group to we want to add a phosphate group to ADP and get ATP. Sorry. So essentially, adding of a phosphate or a phosphoryl group is called phosphorylation. So kinases, essentially how they work. Kinase is typically a large enzyme complex which takes a phosphoryl group and adds it to a protein. Okay, and it usually uses an ATP to do that because that's the source of the phosphate. So ATP has, at the end of it, if we look at the structure of ATP has 3 phosphates normally attached to it, right? Like, again, this is fairly basic, right? But ATP, I'm looking for a good example of ATP, and I guess that's just something to look up so we don't get lost, is this here. And these are the alpha, beta, and gamma phosphates. This gamma phosphate and this beta phosphate are usually indicated in phosphorylation reactions, where the kinase enzyme takes these and places them on an enzyme. And once the enzyme, or places these on a protein, which could be an enzyme, and once these downstream enzymes are phosphorylated, then that means usually that the protein is activated and can essentially elicit a conformational change that allows it to do a bunch of other things. So when we also will take for granted that you all know what protein conformation is, right? So that's super basic in this point. So yeah, we'll kind of skip that. But now that we talked about phosphorylation and essentially how it works, I wanted to talk quickly about enzyme specificity because, again, all of these terms I'm quickly going through because when I talk about phosphofructose kinase, this is going to be really important because phosphofructose kinase activity is actually extremely complicated. It's the most highly regulated step, the third step of glycolysis, and it's regulated because it's a pretty low energy, pretty high negative free energy reaction, meaning it's a committed step, what's called a committed step, that once you're in it, once it's happening, step 3 of glycolysis, it's going to go full force, and therefore, and it's essentially irreversible. So once a enzyme process is irreversible, once we know that it's irreversible, then we should understand that the body will want to regulate it highly because you aren't going to go backwards in that particular cycle, glycolytic pathway, to the previous steps. And so remember, in phosphofructose kinase, the regulation step that we're considering, that's the third step of glycolysis. It happens after glucose is isomerized into fructose 6-phosphate, and that fructose 6-phosphate, via the action of phosphofructose kinase 1 and 2, is converted to fructose 1,6 bisphosphate. Okay, this is again, irreversible committed step of glycolysis. So there are many actors at play in the action of phosphofructose kinase 1 and 2, and those actors are, again, the products of the reaction. They're also products of other reactions downstream. For example, ATP is always an actor in these energetic glycolytic pathway reactions, especially this one. The presence of ATP is a high energy molecule, so that will inhibit this process from going forward. Fructose 1,6 bisphosphate creation will be inhibited, but if AMP is present, adenine monophosphate, then that means that's a low energy molecule, and thus the reaction will be excited, right? And so we want to create more energy. That's how you would think about it. So again, these enzymes, this enzyme, phosphofructose kinase, is specific to we don't want to do that. I don't want to make every anything. It's specific to specific molecules, right? Like so, so I'm trying to bring up this chart so it's easy to see what I'm talking about. But there are different kinds of enzyme specificities, right? So there's substrate specificity, there's bond specificity, there's reaction specificity, and based on, so this is kind of a way to think about it, based on whether a molecule interacts with the enzyme will, again, change the enzyme's shape and make it more specifically activated, or have a higher activation potential to another substrate. So once ATP binds to the phosphofructose kinase to inhibit it, then it will lose specificity for its main reactant, which is fructose 6 phosphate. Okay, sorry, all these phosphates and fructoses are all going all over the place, but it will less likely bind to the reactant because the presence of ATP is basically telling the enzyme it no longer wants to do any more conversion because we have enough energy in the cell for its needs. So the shape of the enzyme will no longer fit the substrate fructose 6 phosphate, right? Remember, there's an active site which has to take in this fructose 6 phosphate. So another quick way of, I'm just going to go through another quick enzyme, which is actually probably the candidate enzyme when talking about these reactions, or like the model enzyme. You've probably heard of protein kinase A, right? It has a simpler activity actually, but it's much more ubiquitous in the cell life cycle and everywhere in the body. The protein kinase A does a bunch of things, including activates a bunch of transcription factors, which are important in the immune system, in cellular growth, in cell division, in cell energy production. It's all over the place throughout the entire body, and it's very important that we understand the activity of protein kinase A, and I'll get deeply into that at some point, but for now, just quickly going over example reaction of where substrate specificity is important is protein kinase A. Essentially, it's a protein which is activated by an upstream molecule called cyclic AMP. And cyclic AMP is cyclic adenosine monophosphate, right? So you know about AMP, and then there's the cyclic version, which has again, a cycle here. This is, I guess, the easiest way. There's straight chain adenosine monophosphate and cyclic AMP. So cyclic AMP, and by the way, the actual anatomical reaction which occurs is when the adrenal medulla produces cyclic AMP, when it's stimulated to produce cyclic AMP when you have your fight or flight response, when you have a fear reaction that stimulates that production. That cyclic AMP then gets created, and then that binds to protein kinase A. And initially, protein kinase A is inactive. Cyclic AMP comes along and binds to regulatory subunits of protein kinase A. So these boxes here are regulatory subunits, and then these regulatory subunits let go of catalytic subunits. So basically there's 4 subunits all connected together in protein kinase A. The substrate specific regulatory subunits, which are substrate specific to cyclic AMP, are normally holding on to these catalytic subunits because they are also specific to a certain pseudo sequence on the subunits of amino acids of these catalytic subunits. But once the cyclic AMP comes by to these allosteric sites and activates or reconforms the protein kinase, then it lets go of these catalytic subunits, which then these catalytic subunits can go and activate those transcription factors. So the protein regulation is in favor of activation or creation of downstream products. So the substrate specificity changes, right? Because then when cyclic AMP is unbound later on through another process, which I'm not going to go into now, then the specificity, the conformational change occurs again to the regulatory subunits of cyclic of protein kinase, which then will rebind to the active catalytic subunits, which were formally released to go do their downstream work. So that's, again, a substrate specificity element that we can now take over in our next video, or one of our next videos, to the activity of phosphofructose kinase, which again is like the main thing we're talking about: glucose and glycolysis and glucose metabolism. Okay, so that was a lot. Sorry for packing so much into one video. I hope you enjoyed. And credit to all the people who've put together all these pictures here on Google Images, which for now I'm getting away with not drawing the individual units, but I'll be doing that when I organize these videos a little better. So yeah, thanks for watching. Like, subscribe, and I'll talk to you later. Bye.